Okay, so for our final plenary address this morning, we have Andrew Young from CSIRO in Canberra, and Andrew comes from a background in plant ecological genetics. He's currently director of the CSIRO-hosted National Research Collections Australia, but he's also on the Challenge International Science Advisory Panel, and so our ISAP has an important role in ensuring science excellence within the Challenge and building international connections. So Andrew's going to talk about biological collections, essential biodiversity research tools for the 21st century.
Thanks, Thomas. Nigel, I was tempted to ask you as a question, how cold is it up here? Because the temperature varied a bit yesterday and it looked a bit chilly, but I've gone without the jacket and we'll see how we go. Look, good morning everybody. It's a real pleasure to be here in Wellington to talk with you about the role of biological collections in addressing the challenges that we all have around managing our biodiversity, both for conservation and for use. It's particularly nice to be in Wellington today, given I had a text from my wife this morning in Canberra saying it's -2 degrees, and it's always good to escape that kind of temperature regime for a little while if you can. Now first, before beginning my talk, I'd like to thank Andrea and the Science Leadership Group at New Zealand's Biological Heritage for giving me the opportunity to speak to you today about the role of biological collections in biodiversity assessment and management and research in general. Before I begin talking about that though, I'd like to very briefly reflect on yesterday, which frankly I found fascinating. I think this is one of the most interesting groups of people that have come together cross-disciplinary and cross-cultural perspective that I've seen for a few years. And I was particularly interested in Jan's comments yesterday morning, Jan Wright's comments about perhaps loosening the reins a little on how we think about genetic diversity and genetic structure in managing populations. And that's certainly something which in the Australian context is a hot topic, and the concept of becoming more open to the movement of genetic material and populations is very real. I was also really interested in the conversations around access to genetic material and sourcing that material and who it belongs to and the ethics of using that material and what do we want to know, who wants to know it, and how is it going to be used. I found it very engaging and an incredibly important conversation for us to have, and one which, as Director of Australia's National Biological Collections, is front and centre our business, my business, in thinking about those issues and coming up with sound policy which is going to allow both those an ethical approach, a respectful use of material, some boundaries around what it's used for, while at the same time trying to maximise access for benefit. And that's not an easy set of dimensions to get together in your head, but one which we're only going to work through by working together, I think. Now, in all those conversations over the past day, it's become very clear to me that though I am an expat Kiwi who just likes to emphasise his vowels a bit more these days, having moved from New Zealand in the 80s, and though I do come back here every 18 months to 2 years, I only know about 5% of the people in the room. And the reason is I come back here primarily to milk cows with my father-in-law on a dairy farm in Taranaki, which I was actually doing a couple of weeks ago. So I thought it might be useful, and I just put this slide together yesterday or last night. I think that's going to work, to give you a 60-second potted history of the presenter that's in front of you. And so this is me looking at a plant I've worked on for a long time, a grassland daisy in Australia called Ruidosus leptorhynchoides, or the charismatically named button wrinklewort. It's an endangered species, but it has all the great elements of a good model system. It's endangered enough to be interesting. It's not so endangered we can't work on it. Range of population sizes, range of habitat that you find it in. DNA falls out of it, so it's nice and easy to work on. It stays put. The seed only move about 1 metre, so it's just a great model, and you can grow it and cross it in the glasshouse. I started my research work in the late 80s as a graduate student in Auckland, and published my first paper in 1993, which was some work on microclimate edge effects in podocarp broadleaf forest north of Auckland, and trying to understand how big how far those microclimate edges penetrated into forest and what that meant for the reproductive biology and community structure of the plant populations. The last paper that I've got that's come out is in 2016. Now this is an interesting kind of contrast. I've moved from being a plant ecologist. Now, please don't take offence, ecologists, that you're at the base of the evolutionary tree. That's not the intention, the diagram. I've moved from ecology into population genetics. As a graduate student, I moved to Canada to do my PhD, got involved in population genetics and evolutionary ecology, moved to Australia where I continued to do this kind of work. And now, 3 years ago, I found myself as director of our national collections. So I've sort of moved disciplines, I've moved countries, and I've become more and more involved in collections through that period. And as I said, ended up as director of our national collections. And there's also been a migration in the kind of work I've been involved in, not only in discipline, but the way it's carried out. So this paper, which was published in Gigascience last year, is a metagenomics-based analysis of soil communities at a continental scale across Australia. And I guess what I'd like to emphasise is that here I think I looked at 5 or 6 sites, and I think it was funded, if I remember, by a $500 Royal Forest and Bird Protection Society student award. And I think the whole thing got done for that $500 and about $1,000 of university money, with bits of string, quad rats, and a lot of walking up steep hills in Rodney. This is a $5 million project with about 44 partners, and I did that by myself. No one else was there the whole time. You'd never be allowed to do it these days with HS&E. It's got about 44 people, 25 partner organisations, $5 million, continental scale measurement, 1,500 sites, 90 billion sequences. And I think this trajectory of interest and the kinds of work that I've been involved in really reflects the evolution of our science area in general. As we understand that measuring and monitoring and understanding the dynamics of our biodiversity is an interdisciplinary, large-scale activity that requires big teams of people to really put the picture together. Anyway, that was an add-on, so I've got to speed up now because I didn't have that slide originally. We live in a really biodiverse planet. I think that's a good segue into large-scale biodiversity. And that biodiversity has been delivered to us by about 3 million years of biological evolution. The best guess, probably the best guess, is we've got about 9 million or so, 8.7 million species. That's eukaryotes we're talking about. Probably 6.5 million of those terrestrial, around 2.2 million marine. Don't hold me or the authors of that paper to any of those numbers. It's plus or minus 1 million. We've only named about 2 million of them, so incredible diversity, and we can't say in some ways if it is incredible because we haven't met any other planet that has diversity, but we know at the moment we're alone, there's a lot of it, and the diversity in form and function and the environments that those species inhabit really, to me at least, is incredible. And I like to think of biodiversity. I was talking to the CEO of CSIRO, and our new CEO is a venture capitalist from Silicon Valley. And it was interesting thinking about Jan talking about risk and how we take more risk. And Larry's encouraging, certainly within CSIRO, us to take a lot more risk. And one question is, how do we prepare the public to have an appetite for risk and to know we're going to fail, when failure may well mean species extinction in some case? It's a little bit of an aside, but it's an interesting question. And I was talking to Larry about what our biodiversity is and trying to explain it to him, and I started using the term, it's an environmental widget. It's something that sequesters carbon for us, it cycles our nutrients, it purifies our water, it does all of these things. It does it pretty flawlessly, and it does it at global scale. It also supports our industry. I mean, our tourism industry is fundamentally based, ecotourism, on our biodiversity. Certainly in many parts of the world, it's an important input into forestry and fisheries. Fisheries are interesting because there you have a wild-caught industry that's based on biodiversity. So there the challenge of managing biodiversity and sustainability is really real, and it's a fantastic case study to look at. And we still, use a lot of wild relatives in crop breeding programs today. It's also a real source of innovation and inspiration, more and more. And if we think about synthetic biology, with people looking to our biodiversity for the source of biobricks to bring into synthetic biology programs, but it has been for a long time a source of that inspiration around new medicines, materials, some industrial molecules as well, and what came out of yesterday to me even more is the profound influence of our biodiversity on human and cultural well-being, a deep human link to that diversity that's around us. The other big thing is it's complex and it's connected. And I was trying to say to Larry how complex and how connected, he said. I said, well, I reckon it's much more complex than the space station and it's more connected than the internet. And that he kind of got. I don't have any data to support that at all, but he got it. And this is the kicker, is we can't make more of it. We can't at the moment build a new one. So we absolutely have to manage the one we have. So we need to manage our biodiversity so we can conserve it, and so we can conserve it, and so we can use it. And to me, there are 5 pretty straightforward basic questions that we need to address if we want to have that knowledge, if we want to be able to manage and use our biodiversity well, conserve it and use it. What have we got? Where is it? How did we get it? How does it work? And what are the threats? Or how does it respond to change? And I would argue, as Director of National Collections, that biological collections can help us immediately with the first 3. What have we got is taxonomy. Where is it is biogeography. And how did it evolve is phylogenetics. This is bread and butter stuff for collections scientists. And I'd say we can contribute pretty well as well to how does it work and what are the threats, the areas of ecology and conservation biology. So what do collections look like as biodiversity data? Well, they're pretty staggeringly good, I would argue. Very broad range of organisms in collections around the world. They're verifiable physical objects, and this is an incredibly important thing. And we heard, Nigel did part of my job for me, thank you. We heard him talk about the value of pathogen collections. I think he said isolate collections sequenced and available. And the nice thing about collections is they're verifiable physical objects. You can go back and look at it, you can go back and remeasure it, you can go back and rename it, you can go back and resequence it if you're not sure. The data are usually spatially explicit. Whoever's collected them has told you where they've got them from. And they're often associated with really rich contextual data about environmental condition and associated species. There's often a lot of cultural information. And I would argue collections are actually some of the most up-to-date registers of biodiversity status. They're very dynamic things, and not everybody recognises that. We send about 50,000 specimens in and out of our collections every year to partners all around the world. These are dynamic research tools, swapping collections with other major collections, and we probably bring in 150,000 to 200,000 new specimens every year. So they're dynamic, they're up to date. And I'd argue they're the original biological big data. And this is where we're going to have a challenge. I'm mildly pathologically addicted to novelty, and I'm going to try and use the question system to ask a question. Now, I thought it worked pretty well yesterday. We had one, look at that, it worked. So this is what you have to do to try and answer the question. And the question I'm asking is, how many specimens do you actually think there are in biological collections around the world? 500 million, that's a big number. 1.5 billion, or more than 3 billion. So I don't know what I need to do now. Maybe nothing. Okay, we've got 30 seconds. So I'll talk while that's going on. All those numbers are pretty big, and And I would argue you could quite rationally, if you don't know a lot about collections, pick any of them. And when I was doing this and talking to colleagues at the Natural History Museum and the Smithsonian as we were bringing these numbers together, I really wasn't sure what it was going to end up being, but you're going to see in a minute. Now, these are only eukaryotes we're talking about. We're not talking about microbes. How did you go? Okay, so a bit of a spread. You're right, it's not 500 million. Is that laser pointer working? No, that was, there we go, sorry, there we go. It's not 500 million, it's actually 4.5 billion specimens in collections around the world. So certainly going for the bigger numbers was correct. How do I move back? There we go, so that's what it looks like. 4.5 billion specimens, about 1.9 million species. This is an estimate, obviously, and those are some of the major collections around the world. And you can see for New Zealand and Australia, we've got about 75 million specimens between us. So it's a fantastic resource. What are these collections currently used for? There was a great report commissioned by Your Royal Society a couple of years ago, looking at taxonomic knowledge benefits in New Zealand and looking at the status of your national collections. And they pointed to a whole range of things that collections are used for. I've just picked out two here around biosecurity and conservation. So they're already being used in all sorts of ways. But in the 21st century, I'm here to argue that collections can do much, much more than this. And probably there have been really two big game changes in the last 15 years that have made a big difference to what we're capable of. The first is the digital revolution. So, high throughput, large-scale digitisation of our biological collections is incredibly important. It's got to be done in a structured way. You don't want to start top left and go to bottom right. And it's important for a range of reasons. First, we can secure a lot of the data which are currently, or specimens that are currently degrading, and the information associated with them. So we all have ageing slide collections. They're probably one of our biggest problems. And photographs and also sound libraries. Quite a lot of the wildlife collections have big sound libraries on tape, and they need to be secured. But the real winner here is in mobilising the collections data and exposing them to the world of research for use. There's been a big effort lately around crowdsourcing of databasing. So I'll ask you another question in a minute. Databasing these collections, just having it in the collection isn't helpful. It needs to be databased, and having that done is an enormous challenge, data management challenge. Exposing the collections through taking a high-resolution photo, getting it up onto a website, and getting crowdsourcing of the information associated with that specimen is proving a powerful way of us rapidly increasing what we can do in terms of getting the collections databased. And if you're interested in that, go and have a look at the Atlas of Living Australia and the Digivol site that's run by the Australian Museum, which is absolutely world-leading technology for doing that kind of work. I would argue, despite that being important, the most important part is the role that digitisation plays in integrating data both across collections so that you can look at your data from the data from one collection with the data from another, but also being able to take collections, biological data, and integrate that with other data layers like climate and vegetation and soil, opening up huge modelling opportunities. The other thing that's going on is automated analysis, so species discovery. There was an interesting paper published a couple of years ago about whether we can actually name all the species we have before they go extinct. And so automating some of the analysis around species discovery once we have image specimens is a current area of active research. And high throughput digitisation is impressive. I don't know how many of you have seen these kinds of technologies. I was in the Netherlands last year looking at the work being done by Picture-A, and this is one of their high resolution cameras combined with a conveyor belt that's cranking out 5,000 herbarium sheets a day. They run 3 at a time, so they're doing 15,000 a day. Now, we actually got a price to come and look at our national collections, our herbarium. The price point's amazing, $1.50 to $2 a specimen. The herbarium's about 1.3 million specimens, so for $2.5 million we could do it. I'd sign a cheque tomorrow. The challenge for us is specimen handling. We can't handle the workflows. We can't get 5,000 specimens out of our collection and back into it every day to feed the beast. So we need to look at some alternatives. So another quick question, what percentage of specimens, because this is crucial for them to be available for research, do you think around the world actually are databased of those 4.5 million? Less than 25%, 50%, or 75%? It's my last question, I promise. This is probably one of the biggest challenges that we face is how do we digitise sensibly? It's not just a technology issue, it's also a strategy issue. Do we prioritise rare and threatens? Do we prioritise type specimens? We're already doing that to a large degree. Do we prioritise species of economic value? Obviously deteriorating specimens is a way to go. But exactly how we do it and how many do you do, and do we want the ability where possible to have population level samples digitised? You'll probably get this one. Yes, I fear that's true. We don't have much of the collection digitised. All right, how am I going for time? All right. Okay, so the second one is the second sort of revolutionary the big game changer is Museumomics, or genomics on museum specimens. Now, I'm not going to go into a lot of detail here because we've got a whole session, or 2 sessions, I think, on genomics over the next couple of days. And we've already heard some fantastic examples of the kind of work that can be done using historical specimens. But I've tried to break it down into what I think are the key things that this can deliver. And when it really comes down to it, it's sort of I think there are four main bits. There's that are driving it. We're getting better DNA from worse and worse specimens all the time, or we're finding better ways to assemble the kind of crappy reads we get, and that's making a lot of difference in terms of what we can access in the museum. Generally moving from single locus to whole genome, and again we saw examples of that with Nigel's talk, moving to things that were sequenced for 6 or 7 genes through to full genome analysis, and that's increasing the power of what we can do enormously. Few individuals to many, and single species to communities. We've heard a lot about eDNA in Fiona's talk this morning about using metagenomics to try and characterise communities. So those are some of those uses. Collections as reference data sets for eDNA. Again, there was some discussion of that this morning and yesterday. You can have as much eDNA as you like, but unless you know what the sequence is linked to, and it's been looked at by somebody who knows that organism, that group of organisms, and could determine the specimen, then you just have noise. You don't have data. Using collections as historical baselines, we saw that this morning, an incredibly important role. Using collections as environmental samples, and I'll talk briefly about that. Here's a blue-banded bee with enormous amounts of pollen on its legs. This bee's from the collections from the 70s, and so we can actually look at pollinator by using eDNA, look at pollinator networks historically as well as in contemporary time. And using collections as genetic warehouses, and I talked about collections as a source of inspiration and innovation, and I'll give a couple of examples of that. So what is the crazy and ambitious idea? And so I'd ask you to forget everything I've said and then just imagine that we had a data set, and when you're thinking about doing ecology, evolutionary biology, population genetics, the 3 things you really want are a phenotype, a genotype, and environmental information. So imagine you had a data set that had those 3 key characteristics, that you had 4.5 billion data points covering the planet and stretching back in time 300 years, which is what the collections do in many cases. Our oldest collections are back to the late 1770s. And then imagine that was digitally accessible and it was integrated with all the other kinds of environmental data sets you like, climate, land use, soil type. Wouldn't that be fantastic? So my crazy and ambitious idea, and one held by the directors and science directors of many collections around the world these days, is a genomically enhanced and a digitally enabled set of global biological e-collections, because this would be a research tool of enormous power and with an incredible range of utility across a whole variety of sectors, from biosecurity to epidemiology, to biodiversity conservation, evolutionary ecology. So if we had one, what would it do? It's easy to say we should have one. And I'm just going to give some quick examples of what's happening now in the national research collections, the collections obviously I'm most familiar with. I've tried to pick a range of things. I'm going to do these in 1 minute each. It's just me between you and coffee. So national research collections, we might skip over this very quickly. 6 biological collections, the main 6 ones in Australia, 15 million specimens. So that's fish, insects, algae, tree seed, plants, terrestrial vertebrates. They're Australia's biggest set of collections, and they go back about 200 years. So example 1, biogeography and conservation planning. So this is all about data integration and digitisation. So our fish collection, 150,000 specimens, about 3,000 species, so that's probably 60% of what you find around Australia. The nice thing is it's fully data-based, and all of the other fish collections around Australia are data-based, and you can get at the data in an integrated way. And those data have been used to do the bioregionalisation analysis behind the planning for our Australia's new-ish marine reserve system. That's the bioregionalisation there. The fact that the bioregionalisation work and the subsequent marine reserve system was done using collections data gave it enormous credibility, and without doubt made it easier as a policy to roll this out, both, as I said, in the fishing industry, with fishing industry people and with conservation folks, because they believed the data, because their data were there, they were verifiable, it was an object, it was all available. It was collected by biologists who knew what they were doing. And so access to those kinds of data and the integration of the data across the collections allowed us to do this well. They also built the fish map tool, which I can talk to you about later, but also speaks to digitisation around having high quality images of all of our fish and the associated contextual data. Second, pollinator networks, eDNA. So here the Australian National Herbarium, 1.3 million specimens, is being used as part of this project, and the questions being asked are, what do pollinator networks look like, and how do they change in space and time? And pollination is one of the most important ecosystem services we have. You know, there's the standard number that 1/3 of everything you eat comes from a pollination event. So the collections are providing the genetic barcodes for all the plants so that we have the information we need to be able to do the eDNA work, and we're collecting pollen loads off pollinators in a range of communities up and down the East Coast, or we will be. This project's just starting. And then the insect collection gets involved, because we can also then do temporal analysis for some of the pollinator networks, like the blue-banded bee I talked to you about before, who has simply beautiful pollen on its legs. These are the collections from the 1970s. Because we've got those extensive collections, we can go in and do the eDNA work on those pollinators and build historical pollinator networks, and then compare those with our contemporary ones that are using the same technology to build, and look at effects of climate change and land use. Look at that pollen. It's just amazing. I was talking to a colleague of mine who's not that far from here the other day over a beer, and we both got desperately excited about it. It was kind of sad, really. I think the phrase "should get out more" was bandied around a bit. All right, sorry Thomas, I'm moving quickly. So genetic warehouses. Here our algal collection is a really good example. About 1,000 strains, 200 living collections. It's based in Hobart. If any of you get there and would like to look at it, give me a shout and I'll organise it. It's one of our most amazing collections in my opinion. And algal strains have always been, or algae have always been, a rich source of strains for commercial use. And we're working through our algal collection, characterising it and screening it in terms of growth rate and production of a range of bioactive compounds. A lot of that work is done with industry, and I think using our biodiversity is really important to conserving it, because nobody conserves a resource more than somebody who gets something out of it. The obvious story is probably the omega-3 polyunsaturated fatty acids here, which normally you get out of fish, and we found that you can get out of algae, and it's used as food supplements and food additives. Number 4 out of 5, my personal favourite, because I work on wild radish. historical genetics and genomics. So wild radish is a terrible weed. It's not so bad here. I was visiting my dad in Kapiti about 2 weeks ago, and he has a nap during the middle of the day. And I started walking up and down the coast looking for wild radish, and you have a little bit, but it's not nearly as bad as we have it in Australia. So about 200, in truth, more like $300 million per annum off the wheat crop. It both reduces the yield in the wheat crop, so you get a nice wheat crop of 4 tonnes a hectare, it'll go down to half a tonne a hectare if you've got wild radish in it. And then there are also associated costs around contamination of the wheat when you get it. It gets downgraded for cattle food because it's contaminated with mustard seed. So we're interested in how did it invade, where and when, and you can see that we used historical collections there to plot those invasion pathways and patterns. And then the genomic side of it is we'd really like to know where it came from, and we can do that using phylogenetics. We're interested in whether it's a primary or secondary invasion via the US, and whether it's got single or multiple origins, and that's really important. And we saw what looked like a structure analysis, I think, earlier today, and we're doing some structure analysis work around populations. Some are very vigorous and some aren't. And one hypothesis out there is the populations that are really bad are the ones that are getting a lot of benefit out of heterosis. So strains from Europe that have been separated in different countries for maybe 5,000 to 10,000 years are brought together in a farmer's field in WA, and you get enormous hybrid vigour. So we're investigating that. Herbicide resistance has now evolved several times. A lot of these populations, you spray them and they say thanks for the water. So we're now using historical radish collections which aren't herbicide resistant, again, to do the comparative genomics to try and understand the evolution of that trait. Very last one, new kinds of e-collections. So what we have at the moment is mostly physical collections, and we're bringing them into the digital world. Some of our new collections are really only going to exist digitally, and this is probably the best example, and it's that paper that I pointed to in my introductory slide. The Biomes of Australian Soil Environments project. It's a large collaborative project using metagenomic, amplicon-based metagenomics to try and characterise the microbiome of the whole Australian continent. It's been powerful because it's got lots of partners, and sampling was the big challenge. So we have a distributed sampling network with all sorts of different people doing the sampling for us. We send them sampling kits and videos, we go and train them, and then they go and take samples. But there's centralised sample processing around the soil chemistry and the genomics, and that keeps the data quality nice and high. And we also have centralised front-end bioinformatics. And then we have what we call coordinated open access. Anybody can use it. You can get on the website tomorrow yourselves and use it, but you have to request the data. You'll always be given them, but the cost of the request is you have to tell us what you want to do with it. We won't stop you doing anything you like, but we will point you at other people who've made the same request, and we at least ask you to talk to them. What we found is people nearly always do, which is fantastic. This is what the sampling looks like. We've now got 90 billion amplicon sequences. We only have 2,200 frozen soil samples. So this is a situation where the E part of the collection is much, much bigger and much, much more valuable in many ways than the physical part of the collection. It's being used for all sorts of things. The key part was, look at the field work we did. It was just fantastic. It was one of the best projects I've ever been involved in, both in terms of being useful, I think, and in terms of seeing a bunch of Australia. So that's the talk. I hope I've been able to give you some insight into what I think, and many of us think, biological collections to do in the future, and why they're not static resources and museums. They're dynamic research tools with a great future that's being mediated by the introduction of exciting new technologies. But there are challenges, and I'll leave those up there for you. But the one that I think is probably the most interesting one for us to deal with is the last one, which we had a lot of discussion on yesterday, around how do we provide equitable access and use. And the Nagoya Protocol provides us with some vision as to how we do that. But as you can see, there are big gaps in the countries that are signed up to the Nagoya Protocol, or even if they are signed up, they haven't ratified it. Thanks very much, and sorry for going a bit over time and keeping you from your coffee.
Just time for one quick question before coffee.
Thank you.
That was a very engaging, interesting talk. You said we only have one biodiversity for this planet. Imagining that we would take that to another planet, would you think or would you consider that if we move all this digital and lose the actual physical samples, especially in plants, that would be a threat?
So you've talked to my deputy CEO then, who asked me that question. Look, I think the answer is it is a problem if we do that. And the reason is, and we have historical evidence, that we could digitise before we could do good genomics. And if we're digitised, we wouldn't be able, and thrown the specimens away, we wouldn't be able to do the good genomics because we don't have the physical specimen. And we don't know what is the next thing we're going to do. And we've just got a new scanner at the wildlife collection, which is collecting fluorescent spectral analysis of parrot wings, because parrots use that apparently to size each other up in terms of, whether they're good mates or whether, I don't know exactly how they use it and what for. And so that's another set of data which we're now layering on top of the specimen. And again, if we didn't have the physical specimen, we couldn't retroactively go back and collect that new layer. So I think the key with collections is the specimen. It's the point of truth for the name, which is incredibly important. And it's the source of a whole bunch of new potential data layers as technologies come along. So I think digitisation is complementary, but it does, and provides security, but it doesn't replace the actual collection itself. Thanks, Thomas.